Hi everyone, this is your biology class. In this video, we are going to discuss about the enzymes. Used in biotechnology. First enzyme, we will discuss the lysine enzyme. We have to isolate the desired gene from the cell. And we know gene or DNA are present inside the nucleus. So first we have to cut open the cell through which desired DNA is isolated. If we are isolating the DNA from plant cell, we know in case of plant cell there is present cell wall, and then after cell wall cell membrane is there. So to dissolve the cell wall we need two enzymes. First one is cellulose and the another one is pectinase. Both these enzymes help in dissolving the or digesting the cell wall. After the cell wall, cell membrane is there. For dissolving the cell membrane, we need proteases to digest the protein and lipase enzyme to digest the lipid. So in case of plant cell, if we isolating the desired gene or DNA from plant cell, first we have to dissolve the cell wall and cell membrane. For dissolving the cell wall, we need two enzymes, cellulase and pectinase. And for dissolving the cell membrane, we need proteases and lipase. After that, nuclear membrane is present, and for dissolving the or digesting the nuclear membrane, we again these two enzyme proteases and lipase. After digesting cell wall, cell membrane and nuclear membrane, we can easily isolate the DNA from the plant cell. And if we are using the animal cell, cell wall is not there, only plasma membrane and nuclear membrane is there. And for dissolving plasma membrane, again we needed proteases and lipase. And then we isolate the desired DNA. If we are using fungal cell and we are isolating the desired gene from fungal cell, there is a cell wall made up of chitin. For dissolving cell wall in case of fungal cell, we need chitinase enzyme. And then for dissolving plasma membrane, we need protease and lipase enzyme. If we are isolating the DNA from the prokaryotic cell, for example bacteria, in case of bacteria, cell wall is made up of peptidoglycan. We know for dissolving bacterial cell, lysozyme enzyme is present. So we cannot use the other enzyme for dissolving cell wall. We use the lysozyme which dissolves the bacterial cell, making a hole in it, and we can isolate the desired DNA from the bacterial cell. So these are the four lysine enzyme. It depends on the cell from which we are isolating the DNA. These four types of lysine enzymes we use for isolating DNA. Second enzyme we are going to discuss here, that is endonuclease. It is a very important enzyme in case of biotechnology. We are utilizing it. Endonuclease is also known as molecular Caesar. It cut the DNA molecule at the specific site. So we called it as molecular Caesar. First we will discuss how the endonuclease enzyme are isolated and discovered. Before this we will discuss, in case of prokaryotic cell, in 1963 scientists find out in prokaryotic cell there are present two natural defense system. In case of prokaryotic cell, there are present two natural defense system. That is, we called it as two complex system. One is methylating agent, which insert methyl group, and second is endonuclease. Better we called it as restriction endonuclease. Here I am going to draw a bacterial cell. This is the bacterial cell. Inside the bacterial cell, there is present circular DNA. If this cell is infected by bacteriophage, this is the bacteriophage. Bacteriophage transferring its genetic material DNA into the bacterial cell. This is the circular ring of DNA of bacterial cell. Inside this bacterial cell, there are present two defense system. One is methylating agent and other is restriction endonuclease enzyme. What is the function of restriction endonuclease? For example, this restriction endonuclease enzyme in this bacterial cell identify the specific site, that is where 80 nitrogenous base are present, and it cut the DNA between the A and T site. If AT are present on this DNA, which belongs to bacteriophage. Bacteriophage insert this DNA into the bacterial cell, and here are present A and T nitrogenous base on this DNA. Then this endonuclease enzyme make a cut on this DNA between the A and T and restrict the growth of this DNA inside the bacterial cell. In a similar way, if A and T present on the DNA of the bacterial cell, this restriction endonuclease also make a cut on its own DNA. It will not differentiate between the main DNA of its own cell or the DNA coming from bacteriophage. It makes the similar cut on the DNA of the bacteriophage and DNA on the bacterial cell. If this restriction endonuclease enzyme cut its own DNA between the A and T, so it becomes suicidal. So what happens? This methylating agent insert methyl group on the nitrogenous base, either on the A or on the T. So now this restriction endonuclease enzyme do not recognize methylating A and T sequence and do not make cut between them. So methylating agent, what is doing here? It inserting the methyl group either on the nitrogenous base A or T and restrict the working of endonuclease enzyme. So what are these two natural defense system are doing? Methylating agent is present here and it insert the methyl group on the nitrogenous base, so the endonuclease restriction enzyme is not able to make the cut its own DNA. In this way, they provide the protection to the bacterial cell. Next enzyme we are discussing here is the alkaline phosphatase. That is the third enzyme, alkaline phosphatase. This is the circular ring of DNA, that is plasmid, which is present in the bacterial cell. Restriction endonuclease make a cut on this plasmid and this circular ring of DNA is converted into straight chain linear DNA. It consists of two sides. One end is 3' and the other one is 5'. Towards the 3' end there is present hydroxyl group and towards the 5' end there is present the phosphate group. And we know 3' hydroxyl group combines with 5' phosphate group and forms the phosphodiester bond. Endonuclease cut the circular DNA and again what happens when circular DNA is cut? Linear DNA is formed and further 3' 5' end together join together by phosphodiester bond and it again becomes circular. So here we use the alkaline phosphate enzyme. What is the function of alkaline phosphatase? It removes the phosphate group from the 5' end, and after removal of the phosphate from 5' end, we find the Linear DNA on the 3' end, hydroxyl group is present, and at the 5' end there is no phosphate group. This is the function of alkaline phosphate. So further it does not combine and converting into circular ring of DNA. Now on this plasmid we have to insert the desired DNA. This is the desired DNA we want to add with the plasmid. It also consists of 2 and that is 3' end and 5' end. On the 3' end hydroxyl group is present and on the 5' end phosphodiester bond formation के लिए phosphate group is present. When both these combine, this is the linear plasmid DNA, 3' end, five' end, and this is the foreign DNA we want to add with plasmid. On this 5' end phosphate group is present and on this 3' end hydroxyl group is present. So there is the formation of phosphodiester bond takes place. This is the 3' end. These two fragments of DNA are joined together. For joining these two strands of DNA, one DNA belongs to the plasmid and other DNA is the short fragment of DNA we want to add with the plasmid DNA. For joining these two strands of DNA, we need other enzyme that is known as the ligase enzyme. Ligase is the enzyme which join the two DNA together. Joins the two DNA together. So DNA को join करने के लिए ligase enzyme is required. Another enzyme which we used in biotechnology is the phosphorylase. Which helps in adding phosphate group. So these are the enzymes used in case of biotechnology. First one is lysine enzyme. Second one is endonuclease enzyme. Third one is alkaline phosphate. Fourth one is ligase. And fifth one is the phosphorylase. Now we discuss something more about the restriction endonuclease enzyme. Restriction endonuclease enzyme. How it works. It is known as the molecular seizure. It makes a cut on the DNA at specific site. It recognizes the specific sequence of nitrogenous bases on DNA. There are lots of restriction endonuclease enzyme are present in nature. Every restriction endonuclease enzyme recognizes the specific base sequence and make a cut on the DNA through this specific base sequence. Before discussing this, we understand the palindromes. What do you mean by palindromes? Palindromes is a word which read same in both the direction. For example, in English we use the word Malayalam. if you read from both sides, in this direction as well as in this direction, it read the same. The same palindrome are present into the DNA in the form of nitrogenous base sequence in a slightly different manner. For example, DNA is the double-stranded structure. It consists of the 5' end and 3' end. The specific base sequence is present on the 5' end and 3' end of one strand of DNA, that is GAATTC. These are present on one strand of the DNA. And DNA is the double-stranded. This is the double-stranded strand of the DNA. On this, in the opposite side, C, in front of the C there is present G. In front of the T there is present complementary base pair A. In front of A there is present complementary base, that is T, T, and in front of the G there is present C. So if we read these nitrogenous base sequence from 5' end on this strand, that is GAATTC, and on the other strand we read it again from the 5' end, that is GAATTC. So these are the palindrome sequences. These are the specific site for identification of the restriction endonuclease enzyme. Different type of restriction endonuclease enzymes are present. Every restriction endonuclease enzyme has its own specific base sequences on DNA and recognize it and cut it. These are the palindromic sequence or specific sequence. Here is an enzyme, restriction endonuclease, that is E. coli R1 restriction enzyme, which identifies these specific base sequence or palindrome sequences or DNA, and it cut at specific site, that is it cuts the DNA between the G and A site. Here also it makes a cut at this specific site and identify these specific sequence on DNA. When we cut it, what we obtain? Here is only present G, nitrogenous base. And on the other side, there is present A, T, T, C. And on the lower side, towards the 5' end, there is only present G. And on the other side, long strand of DNA, which consists of C, T, T, A. So when restriction endonuclease enzyme cut the DNA between G and A site, we get the sticky ends or COS sites. These nitrogenous bases are hanging and they consist of the complementary base pair to each other. These are known as the sticky ends. It becomes easier to add the desired gene on this sticky ends. Another type of restriction endonuclease enzyme is also present, which produce the blunt ends. These are the sticky ends. If sticky ends are there, it is helpful in joining the two DNA strand easily. If we insert the desired DNA here, due to presence of the sticky ends, ligase enzyme easily join these two strand desired gene along with the plasmid DNA. There is another type of restriction endonuclease enzyme. For that, another type of palindrome sequences are there, like G, 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 C, 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 5' end and 3' end. On the other strand also, that is G, 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 C, C, C. These are also palindrome sequences, and here the restriction endonuclease enzyme cut the DNA strand between the G and C. So after cutting the DNA strand between G and C, what we observe here, it consists of the triple G, and another side it consists of the triple C. And similarly on the lower side, it consists of triple C, and on the other side it consists of triple G. So their sticky ends do not produce. These are the blunt ends. It becomes slight complicated on the blunt ends adding another DNA strand. So basically those restriction endonuclease enzymes are used which produce the sticky ends. So it becomes easier to join the DNA segments. One more thing we are going to study here, that is naming of the restriction endonuclease enzyme. How is the naming of restriction endonuclease enzyme is done? This is the name of the restriction endonuclease enzyme, that is E. coli ECOR1. The first letter is taken from the genera. Next two letters are taken from the species. And next letter is taken from the strain. And one is the number on which it is isolated. Here E is coming from the genera Escherichia coli, Escherichia bacteria. CO comes from the species, that is coli is the species of this bacteria, and CO is taken in naming the restriction endonuclease enzyme. And its strain is RY13. So R is taken from this strain of bacteria, that is RY13 से R को लिया है, and one, that is it is first isolated from Escherichia coli. In this way the naming is done. One more example we are taking here, Haemophilus influenzae. Influenzae. H is taken from the genera Haemophilus, and from species IN is taken, IN, and its strain is RD. If we take the R from their strain, RV use here in case of Escherichia coli. So here R is not taken, D is taken. It is the D is the small numeral, so we use it as a small numeral here. So we called it as Hind. Hind 1 and Hind 2. Both are discovered. But first isolated, जो सबसे पहले isolate किया गया, first isolated restriction endonuclease enzyme is Hind 2. Hind 1 and Hind 2, they are discovered. Hind first is first discovered and Hind 2 is later on discovered. But Hind 2 is first isolated from Haemophilus influenzae RD strain. अभी तक about 900 strain, 900 restriction and endonuclease enzyme isolate किए गए हैं. But first isolated restriction endonuclease is Hind 2. This is all about in this video. In our next video, we will discuss the vector.